Hi, my name is Rob Atwill. I'm the Director of Veterinary Medicine Extension here at UC Davis in the School of Veterinary Medicine. What I want to talk to you about today is rangeland management of waterborne pathogens from livestock and wildlife. So key points I want you to take away from this presentation is what are waterborne pathogens from livestock and wildlife here in California or across the United States, and to be able to compare that to what do we mean by indicator bacteria. Second big point I'd like you to take home with this presentation is why do indicator bacteria poorly predict the pathogens in our water supply on agricultural and rural watersheds? And then the third main point I want to make is how do we reduce the risk by implementing usually landowner scale beneficial management practices that can reduce the risk. So let me first discuss what do we mean by waterborne pathogens. So there's probably three attributes we look for to really place a pathogen in high importance here in California. First, we really need credible evidence that it's causing human illness. Somehow it's reducing the health of us when we ingest it or exposed to it. The second, it needs to be shed by the animals of concern on the watershed, whether it's livestock, wild animals, whatever the focus is, it needs to replicate in that animal so that it creates enough of a pathogen burden to cause a water quality problem. And third, which is often the most difficult, there needs to be credible evidence that this pathogen is being transmitted through the water route, either because we ingest contaminated water, maybe we're in the backcountry, or because we're swimming and inadvertently ingest contaminated water. So that would eliminate those pathogens that are primarily foodborne, for example, and wouldn't qualify as a waterborne pathogen of zoonotic concern. So classic examples here in the United States would be the protozoal parasites Cryptosporidium parvum and Giardia duodenalis. Classic bacterial examples would be the pathogenic strains of E. coli, for example, E. coli 0157H7. Salmonella, another classic example, and Campylobacter jejuni. A unique feature of each pathogen is it requires a certain dosage for it to infect humans or other animals. So on the slide that I'm presenting, I'm showing how Cryptosporidium has a much more lower infectious dose. Typically, it only takes a couple hundred to infect a susceptible individual. In contrast, many strains of E. coli 0157H7 require a much higher dose in the watershed to cause human illness. So each pathogen has this different attribute of how many pathogens it takes to cause human illness. In contrast, what do we mean by indicator bacteria? Well, typically they are not pathogenic, and they are bacterial species that when present in water or food suggest that there may be fecal contamination of whatever it is we're trying to protect, and as a consequence, possibly there are also pathogens. So indicator bacteria, as I mentioned, are often not pathogenic, but they're at very high concentrations in the feces of ourselves, humans, domestic and wild animals. But these are very old technologies and how we monitor microbial water quality. Many of us are familiar with total coliforms. Many of us have heard about fecal coliforms. More commonly in many regulatory agencies is to use indicator E. coli. But all of these pathogens, or all of these indicators, do not do a very good job in predicting when pathogens are present in a rural agricultural watershed, which I'll explain in a few slides. So E. coli in the generic form is actually beneficial in our gut, and we continue to learn through a variety of new scientific techniques what are the health services that generic E. coli provides us in our gut. Often there are 1 million of these bacteria per gram. Only a subset of E. coli are hemorrhagic or pathogenic, and only an additional subset are E. coli 0157H7, for example. And then an additional subset are the outbreak strain that might have caused a problem last year. So what I'm trying to convey to you is most of the E. coli is beneficial, and only a small percentage is pathogenic. So that begins to answer this question, why is there often poor correlation between bacterial indicators and waterborne zoonotic pathogens? So I'll give you a concrete example that we have published on in years past, working with different livestock owners. And different pathogens. So if you test, let's say 100 cattle, look at their fecal sample, you will find almost all of them are shedding millions of generic E. coli per gram of feces. But in that same set of 100 fecal samples, it can be quite hard to find salmonella. Maybe only 1 or 2 animals will be infected. A couple might have crypto. A few might have Campylobacter. So while all animals shed generic E. coli, a subset are shedding pathogens. So it often sets up the problem of a false positive when we find these generic E. coli. One concrete example would be the classic protozoal parasite Cryptosporidium parvum. So again, our 100 fecal samples, we can find on average maybe 50 million E. coli in a gram or thimbleful of feces. In those same animals, a handful of cattle will be shedding 10 crypto or less. So if you do the math, that's 5 million generic E. coli for every crypto-oocyst in adult beef cattle fecal material here in California. Calves, it's a little bit different. We get higher infection levels of crypto, but still it's several thousand E. coli for every crypto-oocyst. So we have the same problems with salmonella, same problems with Campylobacter, that our pathogens just aren't that common. So if you go to a watershed, collect a bunch of samples over 1 or 2 years, what we typically find is only when there's high concentrations of indicator bacteria do we begin to see a positive correlation with pathogens. And then annoyingly, none of us can tell you exactly why, we get these random hits of pathogens even when the indicator bacteria are quite low. So we sort of jokingly say, let's blame the raccoons who came at night the day before you take your water sample. They've defecated in the riparian area, and they create these random pathogen hits. But to be truthful, we don't really know why it's occurring. So you can see the correlation between indicator bacteria and pathogens generally falls apart. Another recent example, several years ago, we collected water samples all the way north up to Red Bluff in the Sacramento River and down south to Modesto in the San Joaquin River over the span of a year. About a third of these water samples were positive for salmonella, and almost all of them had generic E. coli. So when we dig into the data, what you find is, on average, generic E. coli, there was 460 of these bacteria in 100 mils, about a teacup. But in those same samples, there was less than 1 salmonella on average in that same 100 mil or teacup. So there's about 3,000 more generic E. coli than salmonella in those 2 main rivers. That's what sets up the problem of a false positive. So hopefully in the future we'll develop new technology that'll be more precise and more accurately signal when does a river or a lake have pathogens and not rely on these decades-old technology. So if we want to reduce the risk, if the community decides it's in their best interest, maybe with the support of the regulatory community, the land managers, somehow we all come together and make the decision that the risk is unacceptable for the health benefits, the ecosystem services we want from this body of water. So we need to implement some form of a beneficial management practice. The scientist will still debate, what does it mean to have 100 cattle, 200 goats, a herd of elk up on a hill slope to the downstream community that's using this water? How many extra cases occur a year? We're going to argue and argue, but for sure, I think there's credible evidence that we can implement beneficial management practices that almost consistently reduce that risk, whether it's high risk, moderate risk, or low risk. So we can move forward even though we don't know exactly how many cases are occurring. Unfortunately, at this time, we don't have a magic bullet vaccine that eliminate most of those primary pathogens that I talked about in the earlier slide. And even if we did have a vaccine, it's an interesting social question. Why should a ranch up on the upper parts of a watershed incur all the costs for the downstream benefits of healthy water? Perhaps the community should participate in sharing the cost of all of the land managers that are working on that landscape and generating and influencing the water that comes off of that watershed. But again, those are social questions and not the topic for today. Most of the beneficial management practices that we've designed either rely on time, pathogens generally die over time, particularly in summer, rely on heat, exposing the manure to some form of heat, like the backcountry horseman, pushing manure out from under a tree, breaking up that fecal pile, getting it dried down and exposed to solar radiation. Or we rely on distance, either temporary, permanent fencing, pushing cattle away from a body of water we're trying to protect. So digging in a little bit deeper in what we mean by beneficial management practices, we need to come up with ideas on how to reduce the rate of pathogen loading. For example, per 100 animals, whether they're wild, domestic, livestock. Do we have technology to reduce the prevalence or the intensity? Some cases, as you might hear on a municipal watershed that has excessive numbers of feral pigs, we got to call in the state federal trappers and cull the numbers down to a point where we don't see water quality impacts. Other BMPs will rely on transport. Most of the fecal material deposited by livestock, wild animals, of course it depends on the species, but most here in California deposit the vast majority of the fecal material on the terrestrial part of the landscape. So how does it get into the water in the first place? Well, when it rains, sufficient intensity, if we've saturated the soils, we get overland flow, and that's where we see movement from the terrestrial part to the aquatic part. So what are the BMPs that can take advantage of that to increase distance between the fecal load? And then the third thing is, how do we take advantage of the natural inactivation rate that occurs on rangeland with pathogens when they're trapped in a manure matrix? You see those fecal pats out there if you're hiking around on rangeland. Pathogens are slowly dying on all of those fecal pats. So how can we maximize that rate of inactivation? So I'd just like to say, I can't cover all of the ideas that we've worked on and others have worked on, but there's 2 free publications. Your local cooperative extension advisor can find these publications and share them with you. You can contact me or others in the California Rangeland Laboratory. There's multiple venues to get good information on a long list of these BMPs. So one of the key questions is, well, we'd like to reduce our risk. Which animal is the primary animal that's responsible for the pathogens we're seeing? Be it a salmonella, pathogenic strain of E. coli. You've got too much crypto. Is it the domestic or livestock animals that are out there? Is it our companion animals? Or is it some indigenous wildlife species that's gotten a too high concentration that's somehow foraging or living very close to the waterways we're trying to protect? So there is a lot of data out there that we've generated that compare livestock shedding of pathogens to wildlife. Across the United States, a whole nother set of scientific studies. So there are some regional differences, so I'm going to show you a little bit about the California data. So a study that we've recently completed with UC Cooperative Extension Advisors and landowners across the state was a survey of 20 cow-calf herds that was conducted a few years ago. And what you can see from the data is salmonella was extremely low. So this was great news. Only 4 animals out of 1,400 had detectable levels of salmonella. That's not even 1%. What about E. coli O57H7? Very commonly, cattle are identified as the major environmental reservoir here in the United States. But after this extensive statewide survey, we only found 5% of our cows and 5% of our calves shedding this important pathogen. Most of the infected animals came from 3 herds. Half of the herds all tested negative. So to me, that's also good news compared to the often stated example that, well, we know cattle are the primary source of this pathogen. I'm not so convinced, and I'll show you some data. We did find Cryptosporidium, we do find Giardia, but with new molecular tools, what we discovered is almost all of the crypto and all of the Giardia is not the species that's infectious for humans. Again, that's good news. So I don't say this to excuse bad management on our working landscapes and on ranches, but the pathogens may not be as high as we previously thought in our cow-calf operations across California. Some of the epidemiologic studies have identified that if you have a long calving interval, 2 studies have supported this, your herd is at higher risk of being infected with this parasite. So whatever you can do with reproductive management, the ratio of bulls to cows, however you think your operation would improve and shorten the calving season, it appears that there's water quality benefits, interestingly, from this practice. Because of numerous produce outbreaks associated with leafy greens that have been traced back here to California from E. coli 0157H7. We've conducted numerous studies in central coastal California for this important bacterial pathogen. Feral pigs we have found now in 3 separate studies about 1 in 20 or 5% shed E. coli. A little bit in coyotes, a little bit in our crows, a little bit in our cowbirds. We didn't find much in deer that hunters had provided. We got a little bit in our tule elk, and occasionally we get a hit from our field rodents on rangeland and in these produce fields. So contrast this low prevalence of E. coli we find in wildlife to the 2.5% we found in this study in central coastal California, and you don't see much difference. So that's why I'm saying we no longer believe that beef cattle are a primary source of environmental E. coli 0157H7 relative to wildlife. They're all about the same. They're in low single digits. Salmonella, in contrast, wildlife appear to be 10 to 30 times more infected than our beef cattle. So in this second study in central coastal California, we yet again confirmed that cow-calf operations are very marginally and lowly infected with salmonella. So again, I think this is good news. In the same study, we went ahead and trapped deer mice, which are overwhelmingly comprised of, deer mice. And what we find is not that much E. coli 0157H7. It is a very small fecal sample, so it's actually more difficult to find pathogens when you have a very small pellet compared to a handful of manure. We found about 3% infected with salmonella, but what really, really surprised us was that 30% of these rodents were infected with Cryptosporidium. And when you do the molecular tests, about half look like they're human infective. And if you take the time to count all of these parasites in a small thimbleful of feces, there are millions of these parasite eggs per gram of rodent feces when they become infected. So this is in sharp contrast to the data I showed you on beef cattle, where we did not find human infective Cryptosporidium. So again, I would urge everyone to keep an open mind when we find pathogens in our water supply on an agricultural watershed or in a more rural or wild landscape. Don't immediately think domestic animals. Our wildlife here in California are perfectly capable of contaminating water. So what can we do about it? Well, we can develop, design, and implement beneficial management practices. The big goal, if you can understand how bad it is in terms of pathogen loading, that's how effective of a B&P you need to develop, right? You need to match the efficacy of your intervention to the size of the pathogen load. When it fails, it's often because we've implemented the wrong tool on the wrong landscape, or we don't understand this matching. So I've already talked to you about pathogen loading. It's critical that we know which animal is responsible. How do we interrupt transport from the terrestrial part of the landscape where most of our pathogens are to the aquatic? How do we accelerate inactivation? And let's always make sure that the pathogen we're looking at is actually infectious for humans by high-quality molecular tests. So we've talked about this in the past and have shared this with many of the regulatory community and the landowners here in California. Those of you that, for whatever reason, it fits your business plan for your ranch, you've decided to do fall calving. To the extent possible, keeping that calving interval as short as possible. I showed you that that's associated with the reduced risk of Cryptosporidium infection in your herd. The earlier you can calve out, so that we don't have young infected calves shedding this parasite during the rainfall season. Once those soils saturate up and we start getting pasture runoff, that's when we get problems. I know some of you can't move your calving interval for a variety of reasons, but if you can avoid infective calves during the rainfall season, you're really going to help microbial water quality. One suggestion, if possible, and again, it's not going to work for a lot of folks, is to move to spring calving. If you have access to irrigated pasture so that you can have high quality forage, when that mother cow has very high energy demands, you have a calf dependent on that mother cow, that works great here in California, generally speaking, because we don't get runoff. The key, of course, is don't overwater your irrigated pasture so that it looks like wintertime and you have runoff with pathogens. If you can't move your calving interval and you're dependent on summertime on some type of public land, maybe you're up on Forest Service, you're up on BLM land, kicking your cattle out, they typically have a tendency to move to areas of high quality forage. Well, that just makes sense. We like to hang out by rivers when we're up in the mountains. So do cattle. So you're going to have to think of the classic range science techniques to disperse animals from waterways so that when they defecate, it's not near these streams or standing bodies of water. So where you place your attractants, do you hire someone to push cattle all summer? Yeah, it's going to take work, but to try and minimize animals loafing and defecating in proximity to water. If that's too difficult, then of course we have the big F word, fencing. Yes, it's controversial. Many of us don't like to see permanent fencing on public land because it interrupts the flow, both of us, the backpackers, the fishermen, and of animals. Temporary fencing may be a better option. A classic question is, how far back do we build a fence? So this is actually a very technical question. It depends again on, well, how many pathogens do you have in the manure? The worse the infection is in your herd, the farther back you got to go. Just makes sense. Where do the animals loaf? So do the smart thing. Don't place attractants right against the fence that's right near the creek, right? That just doesn't make sense. How does the site behave in terms of infiltration? Have you tried to keep the soil porous so that when it does rain, most of the water percolates down in the soil and acts as a filter? So how does overland and return flow on your ranch? And of course, as we all talk about climate change, storm intensity definitely has an association with what washes off the ranch. That's just common sense. If it rains really, really hard, is that a problem? Of course it is. Almost all BNPs fail under those circumstances. There's quite a bit of data and projects that have been done using soil boxes. There's pros and cons. The pros are, of course, that you can control the rainfall intensity. We can look at species of grass, density, residual dry matter. We can tilt our boxes. Some of the early findings from these soil box studies is that when we spiked in 5 million Cryptosporidium oocysts in a manure pat, immediately rained on it, less than 1% of the pathogens washed off during a 2-hour rain period, 15 millimeters per hour. If you wait a day, that drops down to less than 1 in 10,000. So most of the pathogens in fresh and aged manure are trapped in that manure matrix. Manure is not like sugar. It's not like salt. It doesn't dissolve. It sits there. So if it rains really, really hard, sure, there's erosive forces that'll remove that top layer, and that's where we get the pathogens from manure. But generally speaking, most of it's stuck in manure, so that's good news. Do we trust the data from a soil box? Of course we don't trust the data. It's hard to reduce the complexity of the world to a small wooden box. But there's been important understandings of the processes of contamination. So the good folks at the Sierra Foothill Research and Extension Center were kind enough to host multiple studies, multiple investigators, Dr. Ken Tate, the local county advisors, other folks helping out in these studies to take real-world grass, real-world soil, natural rain, and to look at buffer widths of very short distances, about 10 inches, 10 centimeters, maybe a yard and 2 yards. We can look at different land slope classes, and then we can clip the grass to simulate different grazing intensity. So what were the key lessons from these series of studies? Well, again, I think it's some good news that the soils and the landscapes in our rangeland, our oak woodland, and our annual grassland appear to be very effective under most storm intensities of retaining the majority of pathogens. It either gets stuck in the manure matrix, as we mentioned, or it gets trapped within the first meter or second meter. So buffer distances of just 10 feet can be highly effective to reduce the majority of pathogens that wash off of a site. But you have to have sufficient infiltration. So if your management on your property has encouraged compaction of the soil, you can see it running off, well, then you're not going to enjoy the benefits that can occur from a site that's been maintained. What happens when it rains hard, as I mentioned? Buffers fail. Yes, that's a fact of life. So be careful where you place attractants. Obviously, they compact the soil and create a runoff opportunity, and there's lots of manure deposition. around our salt licks or the molasses tank, whatever you're doing that's attracting animals to loaf in an area. A lot of these studies, if you're interested in further reading, can be found at the California Rangeland Watershed Laboratory here at UC Davis in the College of Agricultural and Environmental Sciences. The website I have here in the slide, Rangeland Watersheds, UC Davis EDU, maintained by Dr. Tate and his colleagues. So I would encourage you to read up on the studies and see for yourself the credibility of this work. Quite a bit of information. Again, something that we can take advantage of in designing these beneficial management practices is the natural rate of pathogen inactivation. In spring, summer, and fall, many of our pathogens do not survive well in a fecal pad. And we have clear evidence that die-off occurs. Generally, the hotter it is, the quicker they die off. So immediately that opens up the opportunity. Let's say it's October and you're in an area that's being carefully monitored for microbial water quality. Part of your ranch has one or more streams, but you have some hill slope access that is a good distance away. Well, you can graze, potentially, if your streams are only ephemeral, only flow during part of the wintertime, but you have no flow for multiple months during the summer, to remove that riparian forage and use it for the benefit of your cattle herd. But then you need to kick the cattle out of that site, let the sunshine and the heat inactivate for a month or two before the rainy season. Can we perfectly predict when the rains will come? Obviously not. So the more sensitive the location, the earlier you probably have to remove the animals. Yes, if you're up in the mountains, there's unpredictable thunderstorms and other issues that can cause runoff and hydrological connectivity. But I think this is a freebie that nature has given us, that pathogen inactivate, and we can use it in our management practices. Another classic example of using the inactivation rate, if you have irrigated pasture, well, why don't you rest that pasture for a week or two before you irrigate? So you move cattle onto the other parcel. You might need several parcels, and your range scientist, whoever you're using for advice, can tell you how to set up a rotational schedule so that we rest that pasture for a week or two before you flood irrigate, overhead irrigate. Then we dramatically reduce the amount of pathogens in manure because they've inactivated and it's hardened. Another classic problem will be, well, I want all of my irrigated pasture to be green and provide fresh forage for my cattle herd. But if you're generating tailwater flows in summer, well, it looks just like wintertime. You're creating runoff. So you might have to sacrifice the low wind of your irrigated pasture to make sure you don't have runoff, but you really dramatically reduce the risk of waterborne contamination by making sure you don't have tailwater flows at the bottom of that site. You could build a little retention basin and capture that water, or if you're lucky enough to have a natural wetland, to allow that water percolate through. Again, you'll reduce maybe 50% the amount of pathogens that are running off of that irrigated site by holding back that water. So a little bit of data, again, from the Sierra Foothill Research and Extension Center and Dr. Tate's lab. You can see from the data that water flowing off of an irrigated site above the wetland has higher counts of indicator bacteria. It goes through the wetland and it drops substantially by about 75%. And the longer we have let that pasture rest, the lower the bacterial counts coming off that site. So again, it's something that I think that we can implement in a low-cost way, some temporary fencing perhaps, moving the cattle between pastures and allowing that fecal load to inactivate. Don't be a knucklehead and take a backhoe to these small little wetlands. Yes, it's soggy and it's wet and maybe it's bothering the cattle and causing other issues. But if you can reduce the flow of the water through that small wetland at the bottom of the irrigated pasture, it substantially improves the ability of that small wetland to remove pathogens. It'll work for you in improving water quality and reducing the risk. So to sum up, the key to success in implementing beneficial management practices, either on rangeland, rural landscapes, up in the high country, is to match the design and efficacy of the B&P to the local conditions and the expected pathogen loads. Once you get that proper pairing, we generally have high success in these B&Ps. So lastly, I'd like to say to all of the ranchers out there, Thank you very much for collaborating on these projects. We always appreciate it here at the university. It allows us to generate real-world data for real-world problems. And with that, I'd like to say goodbye.
You can find more informative presentations covering ranch water quality topics on the UC Rangelands website, and look for the Water Quality Information Hub or direct your web browser to the URL shown on this slide.